Hello everyone, my name is Jian Kai Yang and I work as a principal scientist and group leader in the Department of Formulation Development at STA, which is a Wuxi Aptac company. And today I'm going to present about the scaled up production of Lumefantrine nanoparticles. Before I jump into my main content, I would like to first of all just introduce about the current situation about malaria in the world at the moment, because lumefantrine is actually a small molecule drug developed to treat malaria. So I've gathered some data from the World Health Organization, specifically from their malaria report published in 2019. As we can see in this figure, we've still got a quite leveling off trend in our number of malaria infections in the past 10 years. And the majority of the worldwide infections happened in Africa, followed by Southeast Asia, and then followed by the rest of the world. But really, the majority of infection occurred in Africa, and the number of deaths resulted by the malaria infection in 2015 was still around 500,000. But really, the worst thing about the malaria situation is the vast majority of death cases were children under 5 years old. So that's really a devastating situation, and that requires us to do something about it. So I think we've learned about the current widespreadness of malaria and its devastating effect on children. So it's really important to talk about the treatment of malaria. So actually, the first treatment of malaria was discovered by extraction from a plant called cornula and called cinchona. The medicine available at that time was called quinine. It was first discovered in 1600 in South America, but within the time frame of about 350 years after the discovery of quinine, we haven't really discovered anything new to treat malaria. So it was not until about 1934 when a German scientist synthesized a new molecule had we got no new treatment to tackle malaria. And this chemical molecule synthesized was called chloroquine. It was actually the most powerful malaria melody at that time. But later on, after the wide use of chloroquine, we've discovered that one of the strains that causes malaria, it was one of the parasites that causes malaria, which is called Plasmodium falciparum. It actually developed resistance to chloroquine, and this resistance was first noted in 1950s and 1960s. It first appeared in Southeast Asia, Oceania, and South America, and later on it was pretty much observed in everywhere in the world. So to tackle the increasing resistance of Plasmodium falciparum, Pfizer developed doxycycline in 1960s, and the joint program between the US Army and also WHO resulted in the discovery of mefloquine. So these two medicines have been both developed to tackle the increasing resistance of Plasmodium falciparum. But up to now, to be honest, all of the available medicines for malaria treatment all have been noted to have resistance from this particular parasite, Plasmodium falciparum. So actually, lumefantrine was also developed in response to this increased resistance, and its development work started in about 1960s. We've got lumefantrine actually as a derivative of artemisinin, and it is used as part of the artemisinin-based combination therapy. It was first of all discovered in 1967. A Chinese scientist, Tu Youyou, she extracted the active compound from a plant called Artemisia annua. This active compound was known to be artemisinin, and then she later synthesized the lumefantrine from this active compound. So that happened in 1967, and in 1960s, the Chinese scientists, they conducted initial clinical study using artemisinin-based combination therapy, ACT in short, and they confirmed the efficacy of the ACT treatment. And then they continued their clinical program all the way through to 1990s, where they decided to run a joint clinical program with a world-leading pharmaceutical company, Novartis. And in this joint program, they undertook an extensive clinical program. So you can see here, this is the number of clinical studies they took, and we've really got a lot of variations of them. So this happened from 1994 all the way to 2006, and finally the ACT treatment was approved by FDA in 2009, and it became in the market under the trade name of Coton. Then the Chinese scientist Tu Youyou was awarded the 2015 Medical Nobel Prize for her discovery of artemisinin. However, due to the hydrophobic nature of lumefantrine, it actually suffers from low bioavailability, and therefore a high dose and a high frequency of dose are often required, and therefore increasing the risk of patient non-compliance. As we can see from the solubility data in 3 biorelevant media, we can see the crystalline lumefantrine has really low solubility, and as we know, patient non-compliance may increase the potential of drug resistance, so we really need to tackle this issue. And our way to tackle the issue is to use the flash nanoprecipitation technology developed by Princeton University. So we'll use this technology to make nanoparticles in a scaled up production mode at STA to counteract the low solubility issue of lumefantrine, and as an aim to tackle patient non-compliance, and in the end hopefully to increase the time window where we can still use ACT as an effective treatment method for Plasmodium falciparum. So before we start, I would like to just give some brief information about the characteristics of lumefantrine. So this is the chemical formula of lumefantrine, and also its structural information. The powder of lumefantrine, the crystalline powder of lumefantrine appears as yellow. It has got a melting point at about 130 degrees Celsius. As you can see from the log P value, which is quite large, it further confirms the low solubility of itself that I was talking about. And here we've got solubility of lumefantrine at room temperature in a range of solvents. It's really not soluble in water, but its solubility in organic solvent is much better compared to that in water. So as I said, the goal here is really to scale up the production process of lumefantrine using flash nanoprecipitation technology, and we want to use the nanoparticles that we produce to demonstrate its potential of increasing lumefantrine bioavailability compared to the commercial counterpart. And if we manage to do that, then we prove a pathway to tackle the potential limitations of the current malaria drugs. So this is the process route that we use to manufacture our nanoparticle for lumefantrine. It all starts with this first step, which is called flash nanoprecipitation, and it occurs in a device called MIVM, which stands for multi-inlet vortex mixer. This has got 4 streams coming into it, 1 solvent stream and 3 anti-solvent streams. The solvent stream contains lumefantrine and a stabilizer, which is HPMC-AS, and they are dissolved in tetrahydrofuran, THF in short. The 3 anti-solvent streams, they are basically aqueous based, and they've got some amount of sodium hydroxide dissolved in them. Then we've got our FMP lumefantrine. After mixing of these 4 streams, after we finish processing the required quantity of Lumefantrine, which in this case the batch size is 300 liter. We store them in a buffer tank, and we will pump them from the buffer tank to a spray dryer to flash evaporate the THF we use. And after we've removed the majority of THF, then we will further concentrate our now THF lean FMP suspension using a device called tangential flow filtration. This is really done to remove the excess amount of water, so that we will make the subsequent spray drying process economical. Before we spray dry, we mix it with some type of cryoprotectant, which in this case is HPMC E3. And after mixing, then we'll finally spray dry to get our dry Lumefantrine nanoparticles. So we've got a 4-step process here to go through: FMP, FE, TFF, and SD. In this program, we've used 300 liter as the batch size of our Lumefantrine FMP suspension, which contains equivalent to 225 API. So in this first step, which is FMP, the size of nanoparticles can be really influenced by solvent content and also the mixing speed. So we've elected to use a solvent content of 10% by volume. As you can see, the volumetric flow rate for the THF stream is 0.25, and the other 3 anti-solvent streams, they've all got a typical volumetric flow rate compared to that for THF. So we've got a 1:10 ratio here, and we are using a molar mass between our succinate group on HPMC AS backbone with regard to our sodium hydroxide as 1 to 0.5. So this was done as a requirement to ionize the carboxylic acid groups on succinate in order to create electrostatic repulsion between the nanoparticles generated to further enhance their stability. The mixing of the 4 stream was done at very high speed, so it's indicated by this Reynolds number here, almost at 48,000. As you can see, by using a 10% THF content at a very high mixing speed, we've got our initial D50 of nanoparticle size at 229, and by using the electrostatic repulsion generated by ionization of carboxylic acid, we've got a very stable FMP suspension. Its stability lasted for 1 day, which allowed us to process them further. So the next step is flash evaporation. As I said, the purpose of it was to remove the excessive THF used to generate the FMP suspension. By removing that, we will reduce the effect of Oswald ripening, so that we can further control the stability of our FMP suspension. The inlet air in the flash evaporator was set to be 90 degrees, so that's enough to remove the majority of THF, but to not boil off our water. As you can see, after this operation, we've successfully reduced our THF content from 10% by volume to about 1.2% by volume, and the particle size was not really changed much by this operation. The D50 still stayed at about 200 nanometer, and because we removed the excessive THF, the stability of our FMP suspension greatly increased. So even after 4 days, we've got almost no change in our D50. Then the next step is TFF. So this process was really operated to remove the excessive amount of water, so that we'll make the spray drying process an economical one. The pore size of the TFF membrane was selected to be 100 nanometer, and you can see we are using a quite high shear rate in our TFF process. So this is really required to shear off any particle deposition on the TFF membrane, so that we won't block the pores. And because of that, we may increase the particle size a little bit because of this continuous shearing effect, trying to joining particles together. But thankfully, to the electrostatic repulsion created by ionization of our carboxylate groups, we're not really increasing the particle size much. As you can see from the data here, we're still well in the nano size range. We've got D50 at about 330, and for our concentrated FMP suspension, we still have a quite good stability as well. So that leaves us with a lot of certainty in using this process to mass produce Lumefantrine because we've got long stability window to operate our process. But there's something that we need to learn from this process as well, which is the permeate flow rate. So for our process, we've used the transmembrane pressure at about 3 bar. This proved to be quite large because it caused the permeate flow rate to decrease quite sharply with time, which is an indication that the pores might get blocked quicker than we expected. So in our next round, we'll propose to use a lower transmembrane pressure, or we'll switch to an even smaller pore size membrane to try to get more leveled off permeate flow rate profile with time instead of this one. So if we have the permeate flow rate approaching kind of an asymptotic value, then we will have a more stable TFF process, and also the possibility to even increasing the batch size without the concern of completely blocking the pores. Then after we've got our concentrated FMP suspension, before we spray dry, as I said, we mixed it with HPMC E3 as our cryoprotectant, and then the spray drying temperature was set to be 120 degrees Celsius. So this was sufficiently high to spray dry our FMP suspension, but also not to cause any melting concerns with Lumefantrine nanoparticles. We've tested the redispersion of the spray dry nanoparticles. As you can see here, the D50 was about 450 nanometer, so it's still well within the nanoparticle, well within the nano size range. And the overall yield and also water content of the obtained nanoparticles was satisfactory. I have to say that this overall yield was calculated from the whole process, so 60% after this 4-step process was not so bad, was not very bad, was still quite high, to be honest. So as one of the objectives of our program was to test whether we've managed to increase the bioavailability of Lumefantrine nanoparticles. So we've decided to test the dissolution of nanoparticles against its against its crystalline counterpart. So I've just copied the crystalline Lumefantrine, its solubility in 3 biorelevant media here, and also the concentration of Lumefantrine nanoparticles from 0 up to 24 hours in one of the 3 buffers recommended by USP. So in this case, we've used the phosphate buffer at pH equals to 6.8. As you can see, we've managed to secure a high level of supersaturation from 0 to 24 hours, which means that we've also got quite good kinetic stability for our Lumefantrine nanoparticles. So before I close up my presentation, I would like to just show our FMP platform and also the other equipment we used to mass produce Lumefantrine nanoparticles. So this is our FMP platform. We've got MIVM sitting at this little ridge here. There are 4 streams, all controlled by mass flow rate controllers feeding into the MIVM, and the output of the MIVM coming out from here. We've used the spray dryer to process our flash evaporation step and also the spray drying step. The only difference between them might be the product from the flash evaporator is a liquid, while the product from the spray dryer from the spray drying step is solid. And between them is our TFF step. So this vertical segment here is the ceramic membrane for TFF, and then we've built a rig to control everything. So we've got buffer tank to hold the feed, we've got diaphragm pump to provide the very high liquid flow required. We've got pressure gauges both at the feed side and also the retentate side to measure the transmembrane pressure, and we've got a control valve here to manipulate the transmembrane pressure as well. So this is our platform for manufacture large scale of Lumefantrine nanoparticles. If you would like to get into use that for other type of nanoparticles, you're also very welcome to contact us. My email address is at the start of my presentation. Thank you for your attention.